Hello, everyone. Good afternoon, people in Europe, and good morning, people in the American continent. So today I have the pleasure to introduce our speaker, Dr. Laurene Tetard. She's a scientist that joined recently the Institute of Analytical Sciences in the University of Claude Bernard in Lyon, France since last year in February as a director of this institute. Formerly, she was a professor in the physics department at NanoScience Technology Center at the University of Central Florida in the USA. So, although Laurene has not worked before in Xylella fastidiosa, she is very interested in promoting some collaborations. Maybe her skills and the technology she applies for her research can also be interesting to the audience. So, welcome to the Xylella Seminar series, Laurene. We will be happy to listen about your research. Thanks.
Thank you for the introduction and for the invitation. I'm very excited to present my work to you and show how some of the work we've done when I was still in the US can relate to the Xylella community, and I hope that will stem some conversation. So today I'm going to talk about my group's work on nanoscale functional analysis of complex systems, and I'm going to provide examples of how this is relevant for plant systems and bacterial systems that could be linked to plant disease, as in the case of Xylella. So over the past 10 years, my group has developed expertise in characterizing some tool to understand complex system at the nanoscale. So in particular, we're working on a platform that is called atomic force microscope, and I'm going to explain how this microscope works in a little while. And we've used that to understand systems that we consider are complex, either because of their chemical composition, their structure, or because they're evolving in time. And so we've done a lot of work on plants and plant-derived products, on bacteria and cells, also on small molecule, on biological filaments that are relevant to cells or even to plants. And because I'm a physicist and I was in the physics department, I've also collaborated with a lot of researchers who work on 2D materials for different applications. But today we're going to focus more on the plant and bacteria work. And when you think of the plant and cross-sectioning the piece of the tree, whether it's a leaf or a stem or the roots, when we explored the tissues at the nanoscale with the tool that is our expertise, what we found is it was very difficult to make sense of the observation we're doing at the nanoscale and make it useful for studies at a bigger scale, which is what plant scientists are interested in. So over the years, we've expanded our portfolio, and we did research on nanoscale imaging, but also microscale imaging with confocal imaging, whether it's Raman or fluorescence. And then we always try to also find ways to work with collaborators to do more of a macroscale or larger scale study and try to link all this information. And I'm going to try to give some example of how we did that on this system. Of course, all the work I'm presenting couldn't be possible without the students and collaborators. And so some of the work I'm going to show has been the work of several PhD students and master students, including we started with Dr. Solomon and Dr. Cost. And then we did a lot of bacteria work with Brianna Lee, and they've all moved on to other positions, but I wanted to acknowledge their contributions here. So today I'm going to talk about atomic force microscopy, and it's what it is and why we're interested in using this tool to study plants and bacteria. And then I'm going to explain some of the limitation of this microscopy and what led us to incorporate another analytical tool to the platform, which is infrared spectroscopy, to complement the information we're getting from atomic force microscopy. And I'm going to give some example of what we could do with this platform. And also I kept one example at the end that I'm hoping will kind of put in perspective what we can do from nanoscale to microscale with the tools we've developed. So to give a context of the scale we're working in, so plants, of course, are at the microscale, and we can make observations by looking at them, but there are more and more details we can get if we're getting down in other scales and we're using new tools. So for instance, if we work at the microscale, we can see a few cells within the tissue. And then if we go to nanoscale, we can see the composition of cell wall layers. And then even with higher resolution, the kind of intricate network of polymers and how they organize in the plant cell wall. So usually we do that with atomic force microscopy because that's the only platform that is available to probe this information. There's also electron microscopy, which you may be more familiar with, but electron microscopy has some limitation for biology because it requires to have a conductive sample and to do more treatment on the samples before studying them. So that's why we prefer atomic force microscopy, where we don't have to do any staining, any conductive coatings or anything like that on the sample. So we really think we're getting closer to the biological information with that platform. And then when you, of course, think where that lands with respect to light microscopy, so you're probing much more finer details. But there's a little overlap. So for instance, for bacteria, you're able to find them with light microscopy, but the details you're going to get from the platform I'm talking about is more at the level of how the membrane looks like, does it rupture, can we look at vesicles inside bacteria and things like that. So atomic force microscopy works this way. So it's a small diving board. So you can barely see it with your eyes. It's a tiny needle that is nanofabricated, and the processes are done so that you can add a very, very sharp needle at the end of this diving board. And this nanoscale tip here, at the very end, you can imagine it's a few atoms in diameter. And so when you approach the tip to the surface of the sample, the atoms of the tip are interacting with the atoms at the surface of the sample. And this is the force here between the two that is going to play the most important role in this measurement. So depending on the force you have between the tip and the sample, this diving board is going to move upward or downward, and you can use a small readout laser, which is kind of like a laser pointer that is aligned on top of this diving board to measure this, what we call deflection up and down. And this motion is recorded in the position sensitive detector, and it's sent to the computer for reconstructing the images. So it's, in a sense, it's very simple. It only has a tiny diving board, a readout laser, and it's very important to understand the interaction between the tip and the sample, because that's what is going to drive all the work we do. So the complexity is more in the understanding of those forces than on the hardware of the microscope itself. So I don't know if it's going to work. I have a small video here to show, but maybe it's...
Change the laser, a normal pointer.
Yeah, that's what I'm trying to find at the bottom. I cannot see. Yes. No, it doesn't want to. Let's, no, here it is. Okay. So In atomic force microscopy, you have a sample that has some morphology usually. The cantilever will come close to the surface. We align the laser on the diving board, like I said, and then you start moving the tip, and this deflection is what you're recording here on the detector, and this is a view of the reconstructed signal. Then you can do that for many lines, and either you get this one profile or you can reconstruct the profile into an image. And there's different modes of operation. So you have this first mode where you're just dragging the cantilever, and then you have this mode where you're tapping the surface. And the reason that not me, that the community developed different modes of imaging like this was to kind of overcome some of the limitation of contact modes. So for instance, in biology, this contact mode has a lot of kind of scratching the surface when you're imaging. So you could rupture the cell or damage polymers and things like that. And so the tapping is more of a gentle approach in imaging method for soft materials. And then over time, they've been more development on the AFM that allows you to kind of adapt to all the properties of materials you want to study. So just to give you an idea, those are some of images that you can get with AFM. So those are some 2D materials like graphene on just a substrate. Those are plant cells. So you see the optical view here, and then it's one of the region here is imaged with AFM. Those are some grains of material that is called perovskite that is used in solar cell. And then this is a carbon fiber where you can see that this structure is very large, maybe 10 micron in diameter, 5 to 10 micron in diameter. But you can see that when you zoom on the surface, you can see the roughness and all the small information that the material has. And then you can study how it's changing under different conditions and things like that. So this tool is very useful because it provides nanoscale resolution. So you can get fine details about your sample. And then it's been developed to take samples that don't need any stain or tagging or any coating to make it conductive and things like that. It can operate in liquid or in other environments. And over the years, it's been developed to provide more than the morphology, so mechanical property, chemical property, and more. But this is still an evolving field. So what chemical microscopy or chemical AFM meant 10 years ago is not the same as what it means today. And so that's what I'm going to talk about in the next part of the talk. So if you look at this review is a little old, 2006, but already at the time, there were many modes that had been developed using the common platform of the AFM, but to access different properties. So for instance, force microscopy. So you push on the material to see the mechanical property of the sample. Kelvin probe microscopy, magnetic microscopy, capacitance microscopy are those modes that are more applied towards physics and engineering. And then there were some modes that were developed to understand the thermal property of material and some chemistry. So if we think about how we perceive the world at our scale, we always use the five senses so we can get a good description of what is around us. And so with the standard AFM, you can get a view of your sample at small scale that you cannot get with your eye, but you can use the platform to reach a view of the shape of your sample. And then you can touch it because you can use the probe to poke the sample. So that tells you if it's softer or stiffer. But it's more difficult to get all the other information like taste, smell, or hearing. So this is some of when I joined the field of AFM 10 years ago, that was a very active field to try to get this chemical information. I wanted to give you an example of what I mean. So if you get a topography image of two entities and they look similar, but from the details you could tell that maybe they're not made of the same material. But if you only have the image, that's all you can say. And so what we want to do is add a way to determine what each flower is made of, if you will. And the way we did that is by combining AFM with another analytical tool that is called infrared spectroscopy. And that comes to complement other developments also that have been taking place in the field of material characterization. So you see there are quite a few tools that are available, but NanoIR here is recently added to this chart, and so it's in the range of 100 nanometer, 10 to 100 nanometer spatial resolution with a pretty good sensitivity of the information, so you don't need a lot of material to get the chemical information. But this tool is the finer resolution, so the AFM resolution, nanoscale resolution of more common known techniques like Raman and FTIR spectroscopy that are infrared spectroscopies. So the way nanoscale infrared works is that it uses the AFM platform and then it combines the platform with infrared illumination. So we shine a laser that emits in the infrared range on the sample right below the tip. So there are different ways of, again, there are different modes that have been developed, and today I'm going to talk about this mode here, where the laser comes from the top side, and as the material absorbs the light, it's going to heat a little bit and expand, and the cantilever that is sitting is going to deflect, like we talked about earlier. And so by recording this deflection as a function of what we're doing with the laser, we're able to get to the chemical information. How this development came about is by looking at traditional infrared spectroscopy. So in traditional infrared spectroscopy, you have an infrared light source that you shine onto your sample, and usually the measurement would be done in many cases in transmission. And on the other side of the sample, you have the infrared detector. And what you do is you're comparing the light that you send to the sample and the light that is detected. And there's going to be some missing wavelength because some of the light is absorbed by the material. And why is that? Is because the infrared light excites vibrational modes in molecules. So you have the symmetric vibration of the chemical bond, you have the asymmetric, and all these different vibration mode, they happen at different wavelengths. So as these vibrations are excited in the system, then the light is used up or the energy is used up for the vibration and it doesn't make it to the detector. So that's the common way of doing infrared spectroscopy at the large scale. And so then some researchers had the idea that if the material absorbs like that, that means it's going to locally heat because the vibration of the chemical mode is going to make the material expand. And because the AFM tip is so sensitive, maybe we'll be able to detect this small vibration or expansion, photothermal expansion of the material. So over the years, this idea has been evolving because at the time when those researchers in the early 2000s, they didn't have access to infrared laser. The technology didn't exist. But the infrared laser technology evolved, so now it's possible to have a laser that is very directional and then you can control the wavelength, you can pulse the light, and you can do many things. So it's much easier to combine that to an AFM. And so the first iteration of this tool was to send a pulse of light and record how the cantilever is vibrating because of the response of the sample. So send a light pulse and detect the cantilever ring down like this. Then you can do a Fourier transform of the signal. You can select which frequency you want to study, and you look at the amplitude of that peak, for instance, the first one. Then you repeat that at every wavelength that you're interested to shine on your sample, and you can reconstruct an infrared spectrum. So that's been confirmed that the infrared spectra you collect that way, they match, they agree with FTIR spectra. The bands are at the right place, at the right position. So you're actually measuring the vibration of the bonds that correspond to what is in the sample. And after that, the technologies evolved to make the measurement faster, more sensitive, to allow chemical mapping by reducing the time it takes to acquire spectra and things like that. And so now it's possible to shine the laser on the cantilever, do the process I just explained by looking at the Fourier transform and monitoring the amplitude. So in this case, for instance, the second peak as a function of wave number and reconstructing the spectrum. Or you can stay at a given wavelength, for instance, that excites CC bonds, and then you can move your tip across your sample and reconstruct a chemical map of your material. So after doing a lot of these developments in my lab, we also started working with commercial systems, and we started exploring some of the applications that would be interesting to touch with this platform. So at the very beginning, the applications were on polymers. So here, polymer scientists were developing block copolymers where you have two different regions. And then by putting the tip on one region and the other, we could see that we detect the differences in chemical composition of the two spectra, even though the size of the region is in the nanoscale range. And then you could select a band, fix the laser illumination at one band, and map to see which polymer is where by selecting bands. For instance, if you select this band, it's only present in the green. So when you excite the sample at this wavelength, wherever you have a high signature, that means this is the polymer that corresponds to the green here that is present. So that's more for trying to figure out that everything works fine. And it also has a lot of use in polymer science. But recently, the work has evolved to a little bit more complex system to study. So for instance, here we worked with researchers at Oak Ridge National Lab who are developing some vitromer materials. So vitromers are polymers that undergo a chemical change as you're heating them, and that changes also the mechanical property. And so we were able to show that if we're acquiring the spectra as we're heating up the sample, we can detect, so you see here it's color coded, so the red spectrum at room temperature, and here the spectrum at the red dot as the temperature is increasing. And so you can start studying how materials behave under different conditions. And in parallel, there have been lots of developments also in new modes of nano-IR spectroscopy. So all of this kind of works hand in hand, and it allows to broaden the range of application for the tool. So one platform I worked on in the past was to combine the infrared laser to excite plant cell tissues and detect the composition of plant cell walls with this platform. So this is kind of like what the signals look like when we develop the platform to try to see what we're measuring and what has a good response. But what is important is that we can do this. So we can take the cross-section of the plant cell wall, We can map it with confocal Raman. So this is an optical measurement. We kind of like fluorescence measurement, but measuring the Raman signature of each pixel and reconstructing the plant cell wall composition. So we can see that there's a layer that is lignin rich here and that is more lignin poor, or cellulose is more prominent here. And then we can image the same region with AFM, but it's a much smaller region that we image. And then if we kind of overlay the topography, first of all, from the topography of the AFM, we couldn't tell whether there's a change in chemistry or not. If we look at the confocal map, we can expect that there's a region that is more cellulose rich and one that is more lignin rich. But at the time, there was no tool to confirm that. And so what the AFMIR allowed us to do is in the same region to get the morphology image and then to excite the cellulose band in the system and see that with the wavelength we're choosing that is meant to excite cellulose over here. Oh, was for lignin, but the image is for cellulose. Then we can have a region that only one of the region responds, so the region on the top left. So that showed us that we can explore the plant cell wall and start getting some information about cellulose and lignin. So then we did a lot of work on plant cell wall characterization, and for that, how to best prepare the sample so that we wouldn't be measuring just the resin, for instance. So a lot of the protocols in the literature, they use EPON or Ella White, different resin to embed the tissues to do microtoming, and that's fine for optical measurements. But when we did mechanical measurement to AFM and mechanical measurement with the AFM and infrared spectroscopy with the AFM, we found that the resin actually is kind of diffusing into different parts of the cell wall. And also that it's very hard to remove it even after different wash. For instance, here in this graph, you see the mechanical property of non-embedded poplar that was sectioned without any resin. So those are the mechanical property for the different cell wall, S1, S2, and so S1 is this here, this layer here, S2 is the larger region, and the CC is in the cell corner here, which the entire region we call lamella, CML. So this is the range of mechanical property we probe when the wood is not embedded. But you see that with every resin, we actually modified the mechanical property. In some cases, the resin diffused in the S2 layer. And in some cases, for instance, metacrylate, it was more like the knife, the microtome knife would smear the resin on top of the tissues. And so when we probe, we are measuring the mechanical property of the metacrylate that's coating the sample, even though it's a very, very thin nanometer thick layer. And it was really hard to retrieve the mechanical property even after washing the metacrylate. So for wood, it's not really a problem because we can section it without embedding it like we see with the control. But it's more of a problem for instance, switchgrass or leaves or roots. So if we wanted to embed those structures, we know that there's going to be an effect from the resin. So we also confirmed that with a nanowire spectroscopy. So we took spectra across all the tissues, and then we did some analysis on the correlation between the signal of the resin, the pure resin, and the signal we were detecting in the plant cell wall. And we found that all of them were present in our sections. But we continue exploring what we can do with the platform. And so some of the things we've done successfully is to study the effect of plant cell wall deconstruction with the AFM, with the morphology and the chemical information. So that's because I was part of a project where we were trying to understand biomass recalcitrance for biofuel production. And so one of the study we did here was to look at a cross-section with optical imaging, and then to take this small piece of material and do pre-treatment in some solvent. Here was THF in water. Take the section out of the reactor and find exactly the same region on the tissue and compare the property of the cells before and after. So those were the cells at their number, cell 1, 2, 3, and the same cell after treatment. And then we see that at the cell corner here with the 3 cells around it, this region is completely modified. Its morphology is changed after the treatment. But we also added the chemistry. So we found that before treatment, there was a lot of CO bond in the system. And then the C double O, so C double O bond, chemical bond, that CO bond was gone after the treatment, and the only thing that remained was that peak here, which is indicative of water in cellulose. So that allowed us to understand what's going on. And then what we did is we worked with molecular dynamics modelers to make sense of what we were seeing at the nanoscale and combine these two theoretical view and nanoscale view to help the analysis of the reactor scale behavior. So that was one of the first study where we started to try to figure out how our small scale measurement could impact the understanding of larger scale measurements that are more standard for the field. And in this case, it was very successful because we were able to pinpoint which mechanism the molecular dynamics were predicting that were the most likely based on the infrared spectra we collected. So now I'm moving towards the work we did on bacteria that are relevant to plant diseases. So the history of this is that I had a collaborator, Dr. Santra at the University of Central Florida, who's designing new materials that were meant to be new antibacterial agents for plants. And at the time, we were fortunate to be funded several times over several years to work on understanding the effect of zinc-based nanoparticles and also copper-based nanoparticle on different plant system for different diseases. So we had one project on understanding the effect of the treatment he was designing on tomato black spot, so Xanthomonas perforans. And we also did a lot of work. We had several big projects on citrus greening to understand CLAS. So here, I'm going to show first what we did for the study of the effect of the treatment on bacteria. So we developed a protocol to incubate bacteria with the treatment that were designed by his group. First, we did fluorescence mapping to see the life death assay and counts how many cells survived to treatment under the condition we were screening. But then we were interested in seeing what IFMIR could do. So this is one picture that represents a little bit. So we could find that the morphology of the untreated and treated bacteria is changing. So the untreated bacteria are rod shaped and their morphology is pretty consistent from one cell to the other. Whereas we could see some deformation and some different kind of different parts of the bacteria form after treatment. And then we could place the tip at different position on the bacteria and collect their infrared signature. So that's what we did here. So the blue one is on the treated cell and the red one is on the untreated cell. So we see that there's a clear difference in this range for the amide 1 and amide 2 signature of the bacteria. And unfortunately, at the time, our system only had a limited range of infrared illumination. So since then, we've expanded our range of measurements, so we can now get infrared spectra from 800 wave number to 1800 wave number. And so we can get more information about the chemistry of the cell. But nonetheless, the data we collected showed that we can detect some chemical changes and morphological changes that are taking place in the bacteria that is exposed to different treatments. And we also did another protocol that I'm not showing here that is showing that the mechanical property of this bacteria are also changing when they're treated with those treatments. So lastly, I want to show this example of the study we did on zinc oxide-based nanotherapeutics. That was part of the CLAS project, and that was a multi-year, multi-investigator project to get to this publication. So at the time, Dr. Santra was developing this zinc oxide nanoparticle that have a shell of zinc peroxide, and that were very small. So their diameter, individual nanoparticles were below, their diameter was below 5 nanometer. And the idea was that they would be systemic as a treatment. So at the time, all or mostly all orange tree or citrus trees were infected with CLAS and citrus green in Florida. And so they wanted to find a solution to inject the treatment inside the tree to try to make it more efficient for the treatment to kill bacteria in the phloem directly. So we tried both root application and foliar spray, and the size of the nanoparticle was such that it should be able to go through the different layer of the cell, whether in the roots or in the leaf, and enter the vascular system of the plants. So that's what we try to show. So we worked with material scientists to understand the property of the nanoparticle. So we confirmed that their diameter is very small. Here is 2.5 angstrom. So there was a range of size, but in average less than 5 nanometer. And also we worked with neutron scientists and XRD scientists to confirm the crystallinity of the structure, the core shell structures, and all of that. So we did a big part of material characterization for understanding the treatment. Then we exposed the bacteria. So in this case, we couldn't do CLAS, so we always used some model systems. And we exposed them to the treatment. So we did E. coli and alfalfa here. And we did CFU analysis, so standard CFU analysis. We studied the effect of aging of the treatment to see if the treatment would stay in the bucket for a long time. Did it keep its efficiency or did it lose its efficiency? So this is what is showing here. And then we also exposed bacteria to the treatments and did the AFM imaging and chemical analysis at the nanoscale of the bacteria. And we show that all the different cells were affected by the treatment and the chemical signature was changing. And so we could tell that the treatments have an effect, but we could also see that the the mechanism of action is different for the zincicide particle than for common treatment like Norodox or Ergoside. So it started giving us some ideas about how to go about this and develop more protocol. Another thing we did at the microscale or the small plant level, more on the plant side, is that we exposed seedlings to the treatments, and then we section different segment of the seedlings, and we use all our imaging techniques to try to track whether we could find signs of the nanoparticle in the vasculature to understand whether the treatment is indeed systemic or not. So we were able to confirm that. So we found some vasculature where we could see the nanoparticles, and when we did elemental analysis, we could confirm that zinc is present in those chunks that we see in the plant. Then we did sectioning of the seedlings, and we did some more elemental analysis for zinc content, and we were able to map the movement of the treatment inside the plant. So here we see that after 6 hours of treatment of the root, the concentration was highest in the stem. And after 12 hours, the concentration is highest in the stem, but it starts making it all the way to the top leaves. And after 24 hours, it kind of distributed all over the plants. That's important because at the time we didn't understand how the movement is happening. And so this kind of measurement, it also allowed us to select when and where to collect plant tissues so that we have more chance to study vasculatures where the bacteria actually are exposed to the treatment. Because if we were to collect the leaf after 6 hours here at the top, maybe nanoparticles didn't make it, and so we're not studying a treated leaf yet. So all this information was very important for us. And so I think the really exciting part of this project was that we really started from the nanoparticle, a few nanometer size. We went to the seedling side and the kind of bacteria culture level. And then we also work with people in greenhouses and field, and they did extensive greenhouse and field trials to show that the same treatments that we studied at those smaller scales, they have to show their impact on the size of the fruits, on the HLB disease rating in the field, and also at the time we also look at canker lesion and show that those different treatments, they have an effect on that as well. So this really took the entire scale from nanoparticle to really nanotechnology, from the synthesis to the characterization all the way to the field. So in conclusion, I've shown that there are lots of nanoscale investigations that can be done on plant tissues, and those investigations, they can bring out some new information about the system. And I've also showed that as we're also working on developing the platform to make them to make the AFM and AFM-IR more performant and more suitable for different systems. For instance, where right now there's still a lot of work that needs to be done on doing these measurements in liquid, maybe improving the time resolution of what we can do. So we could track evolving bacteria or evolving systems that are being treated instead of freezing the system at that time point. But even then, there's still a lot of things that can be done. And especially in my opinion, when we can design some experimental plan where the nanoscale investigation will serve to provide new multi-scale analysis to provide information to other scale analysis at other scales. So like what we did with zincicide. And so our team in the process has developed lots of protocols to study what are those treatments doing and understand what's going on at different parts of the tissues. And they can be adapted to other problems, specifically other plant diseases and other bacteria. And I didn't show here that work that is ongoing, but we also have a project that looks at roots. So we're applying similar tools to try to understand local property of the outer layer of roots as they're being water stressed or being subjected to other stresses. So we're really trying to see how this can apply to all these relevant systems for plant science. That's it for me. I will be happy to answer your questions.
Thank you a lot, Laurene. So now we have time for questions. You can either speak or just write in the chat.